This video is about DNA replication. DNA is a double helix, and it's made of sugars and phosphates along the backbone, and then A's and T's and C's and G's, these nucleotides, adenine, thymine, cytosine, guanine. And we see that A's pair with T's and that C's pair with G's. That is, cytosines and guanines will pair, adenines and thymines will pair. The base pairing is related to the hydrogen bonds that those molecules form. A's and G's are purines. And you can remember that with the mnemonic, all gold is pure. That means that C's and T's are pyrimidines. Purines pair with pyrimidines based on hydrogen bonds. Maybe there's 2 bonds or 3. When DNA begins the process of replication, an enzyme called DNA helicase will begin to split the 2 sides of the ladder along those weaker hydrogen bonds. That creates what is called a replication fork, where the DNA is forking away. The next enzyme will help to mark where the replication process is going to start. And that enzyme is called primase. So helicase, that ASE reminds us that it's an enzyme, and its job is to unzip the DNA helix. Primase marks where the replication process begins. Our third enzyme out of 4 that we're going to focus on is DNA polymerase. DNA polymerase is going to build the DNA polymer, and it's going to do that by adding the correct match to the nitrogen base. So if there's an adenine in the original strand, then the DNA polymerase will attach a thymine. If there's a guanine in the original strand, then in the new strand, the DNA polymerase will attach a cytosine. Once the segment of DNA gets copied by DNA polymerase, notice that it's being copied from the 5' end to the 3' end. That refers to the way that the sugars are running in the sugar phosphate backbone. DNA polymerase always moves in that direction, from 5 to 3. Once the segment of DNA is copied, then DNA ligase can come through and seal those fragments up so that we have one continuous strand of DNA. I have a DNA model here to help with the process of DNA replication, and just to review. Notice again the double helix shape. Here I have these black pieces are meant to show the sugar, and the white is meant to show the phosphate. So it's a sugar phosphate backbone on my DNA ladder. The first thing that's going to happen is DNA helicase will begin to unzip that DNA fragment. So now I've got this separation here by breaking these hydrogen bonds. Then DNA polymerase is going to come along, and we're going to start building these new nitrogen base pairs, and we're going to do it from the 5' end to the 3' end. So helicase is splitting the ladder this way, and DNA polymerase is copying this way. So in that case, I have a black one here. We'll say that that represents cytosine. The red would be a guanine. And here I have a green one, and the green one pairs with the blue, thymine and adenine. So we continue along that route in order to get the correct pairs, just like DNA polymerase would. And we'll add our sugar phosphate backbone here. And then a ligase would come and seal that up really well, so that we have a new strand of DNA that's identical to the old strand of DNA that we have.